Sí, bueno, probando Zoom.
Buenas tardes, yo creo que vamos a empezar y bueno, ya conforme vaya pasando el agua irán llegando las demás personas. Eh, bueno, primero pues muchas gracias por estar acá, las personas que nos acompañan en Zoom y en YouTube. Es realmente un gusto presentarles al doctor José Luis Pruneda-Paz, quien nos visita hoy en el Seminario de Frontiers y desde ayer en la clase con los chicos de la LSG. El, José estudió una licenciatura en bioquímica y farmacia y después un doctorado en microbiología y biología molecular en la Universidad de Córdoba en Argentina, ambos grados. Y cuando acabó su doctorado, se mudó a California, al Scripps Research Institute, a ya trabajar en relojes biológicos en el laboratorio del doctor Steve Kay. Después se cambió todo el grupo a la Universidad de California, de donde a partir de ahí José ya echó raíces en la Universidad de California, porque a partir del 2011, cuando concluyó su postdoctorado en del doctor Kay, obtuvo una posición de profesor asistente en la Universidad de California en San Diego, hasta el 2018, y del 18 al presente ya es profesor de la Universidad de California en San Diego. De los honores y reconocimientos que ha recibido José, me gustaría mencionar dos, que fue el premio de la Fundación Hellman a jóvenes investigadores que recibió en el 2013 por, por su distinción y las ideas creativas que proponía en ese momento y que, bueno, ahora vemos reflejadas en sus publicaciones. Y en el 2023, hace un año, el reconocimiento como un mentor sobresaliente por los alumnos a que ha dirigido a lo largo de su trayectoria en la Universidad de California. Eh, ese es revisor y editor de diferentes revistas que todos conocemos como PLOS ONE, PLOS Pathogen, PLOS Genetics, varias de la rama de plantas. Y también es revisor ad hoc de diferentes fundaciones de financiamiento, incluidas la de FONCIT en Argentina, en la extinta UCS Mexus, que también participó en algún momento en evaluación de estos grants, para la NSF y para también para la Sociedad de Ciencias de Nueva Zelanda. Entonces es muy solicitado para revisar grants. Evidentemente, para el tipo de proyectos que desarrolla José, pues requiere financiamiento y ha sido muy exitoso en tener grants del NIH y de la NSF también. Eh, Me gustaría también resaltar que pues realmente si leo el CV de José pareciera un investigador de la UNAM que cubre todos los aspectos desde eh, difusión de la ciencia, teaching a todos los niveles, licenciatura, maestría y doctorado. Entonces realmente es muy comprometido con la difusión del conocimiento y también tiene alumnos de preparatoria de vez en cuando en su laboratorio. Entonces empiezan desde muy pequeños. Y bueno, finalmente, lo último que quiero resaltar es que José tiene más de 50 publicaciones, todas en revistas de muy alto impacto, y bueno, pues no me queda más que agradecerle a José por haber aceptado la invitación, y pues hoy nos va a hablar de lo que le gusta a él, del tiempo y de cómo las plantas regulan cómo responden a los ciclos de expresión génica en respuesta a diferentes estímulos. Y de hecho, me llamó la atención que la lista de sus artículos dice que está cronológicamente al revés. Entonces, desde ahí, José, muchas gracias por haber aceptado.
Muchas gracias por la invitación, la verdad que es un placer estar acá muchas gracias por la introducción también. La verdad que he disfrutado mucho de la clase de ayer y de todas las interacciones que he tenido también el día de hoy. Pienso dar el seminario en inglés, pueden preguntar en español, pero ya a esta altura yo no hablo ni bien inglés ni bien español y nunca he dado mi tema en español, la verdad, así que lo voy a dar en inglés. Okay. So yeah, so as Gustavo said in the introduction, my lab is interested in understanding how plants ultimately tell time, how they keep track of time. Okay, and this is mediated by a mechanism that is widespread, across species, that is called the circadian clock. And so the idea is that the circadian clock has evolved across organisms that range from bacteria all the way to human beings, in order to, synchronize physiological events in the organism with specific environmental conditions. So the idea is that we have evolved in a rhythmic world, and so all the mechanisms that we perform, that organisms across, the globe perform, are typically rhythmic. And they are rhythmic in a way so that the timing of those rhythms coincides with a very unique time of the day, which is the time of the day at which that particular mechanism can function more efficiently. Okay, so it's a very efficient way to optimize mechanisms, to optimize the usage of energy, for example. Okay, so the bottom line is that this idea that organisms can do and perform functions, physiological and behavioral functions rhythmically, is really widespread. And so the overall mechanisms that we discover in plants can be sometimes applied to humans or mammals and insects and vice versa, right? So the overall mechanism functions very similarly across species, except for bacteria. The clock in bacteria is slightly different. And so one of the main functions of this circadian clock, as I just said, is to essentially synchronize the behavior of an organism. In this case, this is a sunflower plant, with the rotation of the Earth and the periodic changes that the rotation of the Earth will bring up, right? Like the easiest one to see are light-dark cycles or temperature cycles that occur every day. Okay, if you pay attention to this movie, what you're going to see is that the sunflower is moving, the sun is rising on this side of the screen, and so you're going to see that basically the leaves are following the position of the sun during the day. And you're going to see that during the night, the leaves acquire in this resting position. But if you pay attention, before the night finishes, the leaves are going to position towards the side where the sun will rise the following day. Okay, so these plants can anticipate when and where the sun will rise. And this allows plants to maximize their ability to ultimately photosynthesize, right? To obtain energy, and that's ultimately what's going to drive all the functions that the plant can do. Okay, so the main function of the circadian clock is to be able to anticipate or allow us to get prepared for a particular environmental change. And so when we talk about circadian rhythms, so we can measure different types of either processes, physiological functions, gene expression, protein expression, whatever. And what we're going to see if we look at them over time is that we're going to get some rhythmic function. In a wild-type organism, that rhythmic function will have a periodicity. So one full cycle will take around 24 hours. And because of these cycles, basically the peak of activity or expression that we're looking at is going to occur more or less at the same time of the day every day. Okay, and we call these the phase of activity or the phase of expression. So the phase is talking about at what time of the day a particular function is peaking, all right? And the other thing that sometimes happens is that the amplitude of these rhythms can change. So for example, as we get older as human beings, typically our clocks, they tend to dampen and our oscillations are not as strong as when we're younger. And that creates basically very low rhythms, and therefore all the processes that are supposed to be rhythmic, they're not rhythmic anymore, and that can create some issues, right? Fitness issues and so on. Now, one characteristic of the circadian clock is that the clock can drive what is called self-sustained oscillations. And what this means is that what we can do is to see rhythms in an organism that is growing in a constant condition with no changes in the environment. So in a typical experiment, what we would do is to first, what is called, entrain our organism to some rhythmic condition. In plants, we use light-dark cycles. In most organisms, actually, we use light-dark cycles. And then what we do after, you need 4 or 5 days of entrainment, you can change the conditions to a constant light condition. And in a constant light condition, what is surprising is that many functions will keep oscillating as if the environment was still changing, okay? And that means that there is an internal mechanism, an internal clock that can drive these rhythms, okay? And so when we study circadian rhythms in any organism, we typically study rhythms that persist in a constant condition. That's the experimental condition that allows us to separate clock control rhythms from any other rhythm that is maybe directly linked to a specific environmental change. And in the lab, what we do is to use essentially these two outputs of the circadian clock. We can use we can measure the activity of a promoter that is clock controlled. To do that, what we do is to create transgenic plants that carry this promoter fused to luciferase. And so these plants are going to glow rhythmically. And the advantage of using luciferase is that you can look at luciferase in a live organism over long periods of time. So we can look at the luciferase activity in many, many individuals and follow those rhythms, which are going to follow in a wild type scenario, what you're seeing here on the left-hand side of the screen. And of course, we can follow also gene expression, which we can measure by using RNA-seq or qPCR, depending on the experiment that we're doing. Of course, in this case, the idea is that the sampling has to be always at regular intervals, okay? So in plants, the circadian clock plants are extremely rhythmic. The circadian clock controls almost every aspect of physiology and development. And this slide is just illustrating some of the functions or mechanisms that are controlled by the circadian clock. But basically plants, they grow, they develop, they flower, they defend from stresses in a rhythmic fashion. So there are always, more beneficial times of the day for all these different functions. And actually, if we create mutants where the plant is not rhythmic anymore, this is just a cartoon of that exemplifies what happens when we overexpress a key clock component for plants, which is called CCA1. I'm going to name this gene several times, so just for you to remember. But if you overexpress CCA1, you have an arrhythmic clock. And plants that have an arrhythmic clock grown in a rhythmic environment, they, as you can see, they have growth defects among some other defects, but there's a significant reduction in growth. Okay, so there is a benefit for an organism to have a functional circadian clock in order to grow with optimal fitness in a rhythmic environment. Now, why we're interested in circadian clocks, of course, as I mentioned before, on one side of things, the mechanisms that we study can be applied to understand how the circadian clock works in other organisms, but also in particularly for plants, as you probably all know, climate change and the increased demand for crops is posing a significant challenge for agricultural productivity. And in this map, I'm just showing you a recent publication that came up in PLOS ONE, I think it is, that shows the areas of the world where the production of a particular type of coffee beans is going to drop and where it's going to increase. And this, what you're seeing, I'm going to show you in a second, that the areas where it's going to drop are painted in red. The ones where there's not going to be any change are, in the, kind of darker brown here. And where it's expected to increase, they are shown here in blue. Okay. And so what you can see is that there is an expansion of the cultivation areas, to out of the equator. Okay. So, and this is not only the case for coffee beans, but for many crops, we see this tendency. So there's an expansion because we need to produce more. So crops are being produced in areas where before they were not produced. And also because, there's, the hotter areas or the warmer areas of the world are basically imposing a challenge for crop productivity. So crop production is moving outside of the equator. So we need to identify strategies to basically adapt crops to the new cultivation areas, as well as envision strategies that will make crops more efficient and crop production more sustainable. And so why I'm saying all this? Because it has been shown that through several years of domestication of many, many crops, one particular trait that was selected for, not on purpose, but most crops, when they are selected because they are more resistant to a pathogen, or they can produce more, right? Or they adapt better to a particular part of the world. But what researchers have been doing is to start looking at the clock function in those crops that adapt to new latitudes. And what you see, this is an example for tomato, is that tomato species that, the wild species originally evolved from the equator, but as the tomato was domesticated and moved to northern latitudes, what it is observed is that the domesticated varieties have a clock that runs with a longer period. So the clock runs more slowly. And it seems like, this clock running more slowly allows plants to deal with the different photoperiodic changes that are going to happen at northern latitudes, right? So in the equator, there is a neutral or mostly neutral 12-12 lidar cycle. As we move, out north or south of the equator, now there are periods, depending on whether it's in summer or winter, the periods of light and darkness, they change. So it looks like having a clock that runs with a more slower pace, allows the plants to deal better with the new environment. Okay, so imagine that if we can now understand how the clock works, we can try to envision ways in which we can tweak, the clock and either improve the adaptability of crop varieties that are already domesticated, or even very rapidly domesticate new varieties, right, or new crop species. So what we do in the lab is to try to study at the molecular level how the circadian clock works. And to understand the circadian system, this is a pretty complex system, but this is a very simplified version of how the entire circadian system works. So at the center, basically it's based on 3 modules. At the center of the system, we have the circadian clock itself, so the mechanism that drives rhythms. And this mechanism, on one hand, is entrained or receives the input from environmental signals. Okay, so these input pathways ultimately are the ones that set the pace or the time of the clock. Okay, so the clock receives inputs from lidar cycles, from temperature cycles, and many other environmental signals, I would say, that essentially basically tweak the clock to be at the right time. Once the clock is entrained, now this clock will drive rhythms in its output pathways with a certain periodicity or at a certain phase or time of the day. Okay? And so what we are interested in the lab is to understand at the molecular level these pathways, and we are particularly interested on the input side of things to understand how pathogens, how the infection by a pathogen can change the circadian clock function. And I'm going to mention a little bit of this at the end of today's talk. Now, in terms of the core clock, as I'm going to mention in a little bit, what we're interested in is to understand how the clock works in different organs of the plants. The reason for this is that for many, many years, the clock function has been studied by essentially smashing, the entire Arabidopsis plant and looking at rhythms that contain the rhythms of shoots, roots, hypocotyls, etc. And there is, increasing evidence suggesting that the clock functions differently across organs, and this is true for other multicellular organisms. So we are interested in trying to understand what is the structure of the clock that allow us to understand how clocks work in different organs. And of course, in terms of the outputs, we're interested in understanding how the clock ultimately controls growth and developmental responses. So this is how you don't need to pay attention to the specific names here, but this is what many of my colleagues like to call the clock confusiogram. So this is how the clock, this is how the core clock mechanism looks like in plants. Okay, so many years of research, again, using whole plants, have identified a number of components. But here the idea is that the clock in plants, as in many other organisms, is built by several feedback regulatory loops that are interlocked. Okay, so these are multiple feedback regulatory loops where one component is repressing the expression of its own regulator and so on. And so at the center of this network, and this, again, this structure is common to not the specific proteins, but the overall structure of the network is very similar across species. Now, at the center of the circadian clock mechanism, what we find are these 3 transcription factors. There are 2 transcription factors that are called CCA1, which I just mentioned, and LHY, which is a very close homologue, that are expressed very highly in the morning. And these transcription factors repress the expression of the evening component of the loop, which is called TOC1. Okay, what happens is that during the day, there are high levels, especially at the beginning of the day, there are high levels of CCA1 and LHY. Throughout the day, CCA1 and LH2 proteins are degraded, which ultimately release this repression, and they make essentially TOC1 peak during the night, okay, at the beginning of the night. And TOC1 has been shown to work in regulating CCA1 and LH2. Okay, so one of our initial contributions to this central network was by studying this particular side of the loop, because it was known that TOC1 can regulate the expression of CCA1 and LHY, but it was not known whether this regulation was direct or indirect. Okay, so closing this loop was a big question, you know, several years ago. But what I wanted to show you is how we ended up kind of closing this loop. And what we decided to do was to try to identify transcription factors that directly bind to the promoter of CCA1 and can regulate CCA1 expression, and try to see if those transcription factors might be somehow linked to the function of TOC1. And to do this, I'm making a very long story short here, what we did is to create a transcription factor, an array of transcription factor clones. Okay, so we cloned pretty much every transcription factor that is encoded in the genome of Arabidopsis plants. And what we did was to place these clones in 384 well plates, so that in each well of these plates, there is a single transcription factor clone, okay? And then what we do is we challenge this entire library with a construct that looks like this, which contains a promoter fragment of interest, which in our case was the CCA1 promoter, driving the expression of a reporter gene, okay? So if we put this, if we have this scenario in each one of the wells of the array, essentially what we are able to do is to basically screen in pairwise combinations the binding of each transcription factor for our promoter of interest. And of course, we can clone any promoter here. That's why this tool has been very widely used in the plant community to very rapidly identify transcription factors that control or regulate the expression of your gene of interest. Okay, and so once you do once you test for the activity of the reporter, you might imagine that you can get something like this, where these red dots are indicating where the reporter activity is high. Because you know that in there is a single transcription factor, you immediately know which one is the transcription factor that binds to your promoter of interest. Okay, and we did a number of not only we created the genomic collection of transcription factors, but we did a number of improvements to the system in order to create a system that can be fully automated. Okay, so we can do this in a very high throughput manner using a robotic station like this one that can essentially do liquid handling by pipetting liquids into these 384 well plates, right? And multiple, many of these 384 well plates, okay? So I'm not gonna have time to play the movie till the very end. The students saw it all the way yesterday. But the idea here is that we have improved really the throughput and we dealt with a number of technical constraints that make now the assay very approachable, I would say. Even if we don't have a robot, we don't need a robot to do the entire screen, right? The robot, of course, increases the throughput. But the bottom line is that we were able, I'm gonna move out, we were able to identify a transcription factor that I call CHE that binds to the promoter of CCA1. And it turns out that CHE forms a heterodimeric complex with the TOC1 protein. And we were able to prove that actually both CHE and TOC1 can bind to the same region of the CCA1 promoter, ultimately concluding that yes, TOC1 can directly regulate the expression of CCA1. And this essentially closed the loop, the central loop that drives circadian rhythms in plants. All right, now, so as I said, we have always studied the circadian clock function using whole plants, but there is increasing evidence suggesting that different parts of the plant, this is a very young plant, right? So you have the aerial portions of the plant over here, the root is over here. But if we look at the circadian rhythms, okay, in different parts of the plant, this is from a publication that is not from my lab, what we see is that those rhythms don't look identical to each other. So basically, the clock function is different in different organs of the plant. And this is also true in mammals, right? So we have a clock in the liver that it works different than a clock in the lungs and so on, okay? So it seems like it's a common feature of how the circadian clock works across species. And so the question that we try to answer, and is what I'm gonna try to comment more in depth in today's talk, is how the spatial specificity is achieved, and what's the extent to which this spatial specificity ultimately controls rhythmic outputs. Okay, in other words, can we identify, really, how many rhythmic genes are in different organs, and can we try to envision what's the mechanism that drives that rhythmicity? And so to answer this question, essentially, we focus on two major parts of the plant that I call organs. I know that some people don't call organs what I'm going to call organ, so I apologize for that. But we essentially focus on the shoot apex of the plant, and the apex of the plant contains the stem cell niche. So all the aerial structures of the plant are derived from cells that are in that part of the plant. Okay, so we are not looking only at that stem cell niche. The shoot apex is a very complex structure, but we basically looked at the shoot apex as a whole. And so the shoot apex is important for growth, as you imagine, because if you affect the shoot apex, then the plant cannot develop leaves or cannot flower. So it's really important for development, right, and growth. And we decided also to look separately at the leaves. And the leaves, are photosynthetic, or is the place where plants will do photosynthesis. Most metabolic pathways and so on are associated with photosynthesis. So this essentially provides the energy that plants ultimately will eventually use to grow. And so the idea was to try to see first, what is the extent of the clock regulation in these two different parts of the plant, and then try to see how, what might be the molecular mechanism that drives these rhythms. And so what we decided to do is an experiment that looks exactly like what I'm showing you here in the screen, where we essentially entrain plants in LiDAR cycles for 14 days. And then we move plants to constant conditions, as I just explained. And then on the day 3 to 4 in constant conditions, we collected plants every 4 hours during one day. And then we dissected many individuals per time point into the shoot apex portion and the leaf portion. Okay? And we did 2 replicate experiments of these, with the same setup. And that's why, we have these 2 tubes here representing the 2 biological replicates. For the rest of the talk, anything that is in red is going to represent data that comes from the shoot apex, and anything that is in blue is data that comes from the leaves. Okay? And so the first thing that we did is a PCA analysis. The only thing that I want to point out here is that, as you can see here, most of the variance, okay, is accounted for the organ of origin. Okay, so the transcriptomes seem to be very different between the leaves and the shoot apex, as you can see here. What did we find? Well, when we look at the genes that are expressed in one or the other organ, we found that there is a very large overlap, 17,000 genes, and most of them are expressed in both organs. Okay? And then what we can do is to run an algorithm that will look for rhythms, for 24-hour rhythms in this data set, which is called metacycle. And what we found is that there is a large number of genes that are rhythmic. About 5,000 genes are rhythmic. But what was very interesting and unexpected is that although most genes are expressed, what you can see here is that most genes are rhythmic only in one of the 2 organs. So as you can see, we found that the vast majority of the genes are rhythmic in the leaf only, okay, or in the apex only, and only, this is 14% of the genes are rhythmic in both organs. And so the question here was, well, could it be that maybe genes that are, that we find as uniquely rhythmic in one organ, they're just not expressed or expressed at very low levels in the other organ? So we're calling them as being rhythmic in one organ, but it's because they're just not expressed in the other one, right? And so we looked at the expression of all the genes that were exclusively on the red sector, so the apex only or leaf only rhythmic genes, and we look at their expression. And we found that most of them, which are in the light color over here, were actually expressed in both organs. Okay? Not only they were expressed in both organs, but when we look at the ratio of average expression between the shoot apex and the leaf, what we found is that they seem to be actually expressed at very similar levels on average, okay, in both organs. So genes seem to be essentially rhythmic only in one organ, but they seem to be expressed at similar levels in both. So what we thought is, well, maybe we have less rhythmic genes in the apex because maybe the apex has a weaker clock. So you can imagine that if you have a weaker clock, maybe the amplitude of those oscillations are going to be lower in the shoot apex. So we detect less rhythmic genes in the shoot apex because of that reason. And so that's the question that we try to answer next. Okay, so is this difference or this specificity for having rhythmic genes in different organs, okay, specific for each one of the two organs, a function of having a less robust clock in one of the organs? And so what we did here was, when we ran metacycle, we can get the amplitude of all the rhythmic genes, okay? So amplitude, again, difference between the peak and the trough of expression. And if we look at the overall amplitude distributions of rhythmic genes in the shoot apex and the leaf, we find that the amplitudes are actually very similar, okay? So essentially, you know, this distribution of genes being rhythmic only in one organ doesn't seem to be a function of the clock being less robust, or, you know, driving rhythms that are less robust in one organ. And so what we hypothesized at that point is that this looks like more like specific genes are being targeted to be basically down, the rhythmicity seems to be dampened in a gene-specific fashion more than an organ-specific fashion. And so we did a heat map over here. What you're seeing here is a heat map of all the genes that were rhythmic. Okay, on the top you have the genes that were rhythmic in the apex, but not in the leaf. Next, the ones that were rhythmic in the leaf, but not in the apex, and finally the ones that were rhythmic in both. And so what you can see here is that, of course, you know, the genes that are rhythmic show a very, you know, strong, you know, difference between the peak and the trough, which are represented by the black, you know, the darker black, or blue in this case, or red in this case, colors. Now, what you see is that in the organs where the genes were not rhythmic, you know, there seems to be a very residual level of rhythmicity. Okay, these values, these colors, are very close to zero. So the values, you know, the oscillations that you're seeing here are very, very tiny, if at all. I mean, and that's why the algorithm is not detecting them as rhythmic genes. So we were doubting about this. You know, what if we take a different cutoff to assign, you know, rhythmicity, or a different cutoff to assign expression? And so we did. We ran a number of different, the same analysis using a number of different cutoff values, okay, both for expression and for rhythmicity, and the overall results pretty much didn't change, okay? So actually the genes that were called as not rhythmic by the metacycle analysis, you know, had really very, very high p-values, so they were not even close to the cutoff mark, you know, the cutoff value that we selected. So it seems that, you know, in each one of the organs, there seems to be a mechanism that controls the amplitude of specific genes. And so if you can do that, then you can imagine that you can have genes that are going to be, that are not going to be rhythmic in one organ and are going to be rhythmic in the other one. And so, okay, so the next thing that we did was to say, well, if there are rhythmic genes, you know, that are specific to each organ, then the functions, biological functions that are associated to those genes, they also should show the same split or a similar split. And that's exactly what we found. So you can see here, you know, at least, of course, you don't see them. I'm going to point out to some of them. But what you can see is that you have functions that are essentially here on the left-hand side. You have, you know, the functions associated to genes that are rhythmic only in the apex. On the far right, only in the leaves, and in the middle, the ones that are in both. Okay, so you can see that there are several functions that are linked to either the apex only rhythmic genes, or genes that are rhythmic in both, and so on. Now, in the genes that are rhythmic in the apex, what we found for the most part were genes that were associated to chloroplast biogenesis. Okay, and this can be seen as, you know, the shoot apex is where the leaves will start developing, right? So at that point, you know, the organism will need to synthesize, you know, chloroplasts in order to create a photosynthetic tissue. Now, in rhythmic genes that were present in both, okay, in both organs, we found that there were terms enriched for circadian rhythms. So it looks, you know, that was something that was good to find. Of course, these are rhythmic genes. We were expecting them to be associated to circadian rhythms, but we found a number of terms that are associated to photosynthesis, to photoperiodic flowering. So plants, measure time, measure whether the days are getting longer or shorter in order to flower. And so these are important functions, developmental functions for the plant that were found actually in rhythmic genes that are in both organs. And finally, in the leaves, it looks at the leaves as a center for metabolic processes and protein synthesis and biosynthesis of many different compounds. And so the prediction is that there's going to be a rhythmic production of a number of metabolites and proteins and so on that is going to be rhythmic, and that is going to happen in the leaves. So at this point, we come up with the first idea, okay, well, how this might be working. And so what I'm presenting you here is like an initial part of a model that I'm going to be building up as the talk moves on. But essentially, what we have is a circadian clock function that we still don't know how it works in the shoot apex of the leaves that drives the expression of rhythmic genes. Okay, and these rhythmic genes can be either rhythmic in only one organ, for example, in the apex, those are associated to, as I said, chloroplast biogenesis, or they can be only rhythmic in the leaves, mostly associated to metabolic and biosynthetic pathways, and genes that are rhythmic in both organs, which in this case, we decided to exemplify flowering, okay? Now, one thing that we also can measure among all the rhythmic genes is what is the time at which they are peaking. Okay, so these genes are rhythmic, but they are not all peaking at the same time of the day. Okay, in other words, what is the phase of expression for rhythmic genes? And what I'm showing you here, this is called a polar plot that is representing the 24 hours in a day. Okay, so we start here from hour 0 all the way to 24. And then on the outside of this polar plot, what you have here is the light period. Okay, that's why this is colored in white. And then this is the subjective night of the plants. Remember, these samples were collected in constant conditions. So that's why we call this the subjective night, because the plants were actually, in the constant light condition. And what this is represented, this is a density plot that represents all the phases of all the genes that were rhythmic, either in the apex in red or in the leaves in blue. And what you can see here is that the distributions are very similar in this case. These are the distribution for genes that were rhythmic in both organs. So this is the same set of genes, okay? You see that they have the same distribution of phases, but they are shifted, okay? So genes that are peaking in the apex, they tend to peak first or before the peak that occurs in the leaves. And to be honest, we don't know why this happens. We have been, trying to address or identify why this could be advantageous. So if you have any ideas, I would really welcome those. But what we said is, well, this gives us a very general perspective of, this kind of phase shift or phase offset, as we call it, between the peaks, the time at which the genes are peaking in the shoot apex and the time at which the genes are peaking in the leaf. Because these are genes that are rhythmic in both organs, now we can do a gene-to-gene comparison, and we can calculate what's the difference, okay, between the peak of expression in one organ and the other organ for each one of the genes that were rhythmic in both. And what you can see here is that there is, 76% of the genes that are rhythmic in both organs, they tend to peak first in the shoot apex, okay? So there is a very, very significant tendency for the apex rhythmic genes to peak first in the apex. And this is represented by this little cartoon, okay? So basically the idea is that the peak of expression is gonna happen in the apex before the leaf, okay? All right, so what drives these organ-specific, phases and amplitudes that I have been talking about? And so what we decided was that there could be two scenarios. One where we have two distinct clocks. So you can imagine if you have one clock in the shoot apex, one clock in the leaves, they're both working independently. Well, they can drive then gene expression in different ways, right? They can have specific phases and amplitudes to the rhythmic genes that are in one or the other organ. The other idea is, well, we have a common clock, okay? And then what we have are mechanisms that control the phase and the amplitude of oscillations that could be either upstream of the core clock, so modulating how the clock works, or maybe downstream of the core clock, so modulating, the rhythms of genes controlled by the clock that they can be further modulated after the clock has been driving that rhythm. And so we decided to look at all the clock components that were known so far, all of them identified from whole plants, remember, and look at how they are expressed. Are they expressed in both organs or in one, or are they more highly expressed in one organ and more lowly expressed in the other one? Trying to find differences in this network, find whether there could be something that is unique to the shoot apex or the leaves in terms of the clock function. And so the idea was that, if we find something like this in the correlation analysis, well, this would mean that there are unique clocks in either the leaves or the apex. That would be for the first hypothesis. And in the other one, we should find something like this, where there is a high correlation in the expression and amplitude of the core clock genes. So what did we find? What we found is something that looks like this. Here, what I'm showing you is, again, all the known clock components. On this side of the screen, I'm showing you the average expression of those clock components. So how much of them we can find on average across the entire time course. And on this side of the screen, their amplitude. So how big is the difference between the peak and the trough? And what you can see overall, there are a few exceptions, but overall, most or all clock components are both expressed at similar levels in both organs, and they are expressed with similar amplitudes in both organs, okay? So this suggests that there is a single clock, right, that controls rhythms in both the shoot apex and leaves. So we say, well, if there is a single clock, then if we look at the genes that are rhythmic in each one of the organs, and if we look at their promoters, we should find cis elements that have been associated previously to non-clock components. Okay, so we look at sites that were enriched in the genes that were rhythmic in the shoot apex, or genes that were rhythmic in the leaf. And what did we find? We found only two motifs that are enriched. The first thing that you can see is that the two motifs are very similar in both. Okay, so that's a first indication suggesting that rhythms are driven by the same clock that targets the same types of motifs. And the important thing here is that both motifs have been linked before to clock components. Okay, so the first one is called the evening element, which is the motif that is bound by CCA1 and LHY, which I already mentioned, as well as other transcription factors of the same family that are called reveles. And the second motif has been associated to TOC1 and other top members of the TOC1 family, okay? So it seems now, if we go back to our model, that we have a single clock. And so we have a single clock that works very similarly in both organs. How is that we can get different numbers and different sets of rhythmic genes in one and the other organ? And so what we hypothesize is that there should be a mechanism that ultimately brings amplitude modulation that is downstream of the mechanism that actually drives the rhythm. And what this ultimately means is something that could be seen as controversial. What we're saying is, well, there can be genes that are controlled by the clock, but then there is a mechanism in a specific organ that makes that rhythmic gene to be arrhythmic, essentially, okay? And so the next question was, well, how is phase established? What determines the timing, okay, of a peak of expression in each one of the two organs? And again, remember, we could imagine a timing could be here as a clock input, in the clock input side of things. So basically, some mechanism that changes the timing of the entire clock, and that would change the timing of all the clock outputs, or a mechanism that is like the amplitude mechanism that we are proposing that is downstream of the core clock mechanism. And so to this end, what we decided to do is to look at, the peak of expression for all the clock components. Not all clock components are rhythmic, but for the clock components that are rhythmic, what we did is to measure the time of peak in the apex and in the leaf. And we calculated the difference, which is represented here by the length of this bar over here. If the genes are peaking first in the apex, they're colored in red. If they're peaking first in the leaf, they're colored in blue. So you can see already that here, most clock components are peaking first in the apex, which very strongly resembles what we find, among the rhythmic genes in each one of the organs. Okay, so it seems that there is actually a mechanism, okay, that is upstream of the clock that essentially shifts the phase of all the entire clock component, the entire clockwork, and that change is ultimately translate to the phase changes that we're seeing among the rhythmic genes in the shoot apex and the leaves. And this is, the shift is represented here by the two middles of the clock, where you can see that the apex is, more advanced than the leaf, okay? All right, so to this end, what we decided to do is try to, figure out if we can find a mechanism that could explain this phase change. And so we decided to turn again our attention to CCA1. And the reason for that is that one is that we found the evening element enriched among the genes that are rhythmic. That means that there should be a CC1 regulation in there, or CC1 regulating these genes. And the second thing is that it has been shown that the timing of CC1 expression is really key to determine the timing of clock components and determine the timing of downstream regulated genes. And so what did we do here? Well, we went back to our transcriptomic analysis and we looked for transcription factors that would be highly enriched in one or the other organ, okay? So this is just representing that we found, around 270 transcription factors that were more highly expressed in the shoot apex and 32 transcription factors that were more highly expressed in the leaves. And then what we did was to go back to our transcription factor DNA assay. We look at all these transcription factors in our collection, and we tested the interaction of all these transcription factors for, the ability of these transcription factors to interact with the CCA1 promoter, okay, using exactly the same system that I showed you before. And this is showing the results of two replicate experiments, okay, and the lines are showing the cutoff values that we have for DNA binding. And what we found essentially is that there were 3 transcription factors that are consistently binding to the promoter of CCA1, okay? Remember, these are transcription factors that are enriched. In this case, these 3 were enriched in the shoot apex. And so we decided, as you can see, the top candidate of this screen is a transcription factor that is called TCP19. So it's the one that increases the most the activity of the reporter. So we decided to follow up and try to see whether we can characterize whether TCP19 is actually regulating the clock function in the shoot apex. And so the first thing that we did was to see, well, first confirm, is TCP19 really expressed only in the shoot apex? So here in plants, what we can do is to create transgenic plants that carry a fusion between the TCP19 promoter, driving the expression of a histochemical reporter that is called beta-glucuronidase. And that will give essentially, or will stain the plant in blue, wherever the beta-glucuronidase is being produced. You can see, I don't know if you can see from there, but you will have to believe me if not, that there is a very tiny dot, tiny blue dot, where the point of the arrow, or where the arrow is pointing over here. And these are plants of two different developmental stages. Okay, so it seems like TCP19 is really expressed only in the shoot apex and is not expressed anywhere else on the plant. And for comparison purposes, here you have the same type of experiment done with CCA1. CCA1, as we have published before, is expressed widely across the plant. So we confirmed that RNA levels were also more highly abundant in the shoot apex. We created also transgenic plants to look at the protein, and we found that actually the protein can only be detected by Western blot in the shoot apex, but not in the leaves. So really, it seems that this is a very specific or a transcription factor that is expressed really specifically at the shoot apex. And then the next thing that we did, how are we doing on time? Sorry. Okay. The next thing that we did is to, we had observed that when we overexpress TCP19, plants tend to flower much later. So we decided to test if this flowering time phenotype would be dependent on the function of CCA1. So we created plants that carry the TCP19 overexpression construct and the mutant CC1-1 allele. So what you can see here is, well, this is the flowering of wild-type plants. This is shows the delayed flowering of TCP19 overexpression plants. The CC1-1 does not have any flowering phenotype, but you can see that now the TCP19 overexpressors that carry the CC1 mutant allele, they flower, okay, very similarly to the wild type and the single CC1-1 control, okay? So this is indicating that ultimately TCP19, at least in the terms of controlling the flowering time, needs the function of CCA1, or requires CCA1 downstream of it. I'm sorry, and these are the quantifications of multiple experiments, of course. So does ultimately TCP19 play a role in the clock function, right? And does ultimately TCP19 control what we were hoping was that TCP19 would be controlling the phase of, or this timing of expression of CCA1. So the first thing that I'm showing you here is essentially a control in wild-type plants that shows in different ways that the peak of CCA1 is happening earlier in the shoot apex and later in the leaves. So here we're showing you what the RNA-seq data looks like. These are independent experiments where we measured CCA1 transcript by qPCR. And these are experiments where we use transgenic plants that carry the CCA1 promoter driving the expression of luciferase. Okay, so what you're seeing here is luciferase activity in red in the shoot apex, in blue in the leaves. Okay, so in all cases, you can see that there's a shift between the peak of expression in the shoot apex and the leaves. And now I'm going to kind of zoom out to show you how an entire time course looks like. Okay, what you have here is a time course of wild-type plants, and what we have done here is to point to the peak of expression with these, arrows in different colors. And this is the same thing that I've shown you before for one of these cycles. Okay, but this is for a full time course in wild-type plants, and this is what you see when you do the same thing, but in a TCP19 mutant plant. And what you can see is that now the triangles in the TCP19 mutant plant are way more aligned one to the other, the blue and the red. Okay, so in other words, in the absence of a functional TCP19 gene, now this phase shift between the apex and the leaf basically not fully disappears, but is significantly reduced. So we can quantify this, of course, across multiple experiments. So each one of these gray symbols is indicating individual plants, and the bigger symbols are indicating individual experiments. So what you can see here is the phase that we find for the CCA1 luciferase peak of expression in wild-type plants and in the TCP19 mutant plants. So you can see that there is a difference, right? A significant difference of about an hour or more than 1 hour between the phase of expression between one and the other. Now, what is very interesting to see as well is that when we look at the phase of expression of CC1 in the leaves, there's no change, which essentially matches what we would expect if TCP19 is only expressing the shoot apex. It should only affect the clock function in the shoot apex, but not in the leaves. Okay, and this is also interesting because there are some work from other labs that suggests that the clock function in the shoot apex is influencing the clock function in other organs. Okay, and what we are finding is that at least by, for the relationship between the shoot apex and the leaves, it seems like this relationship does not exist. So you can change the clock function in the shoot apex without changing the clock function in the leaves. And we can measure that, this is just measuring the delta between the shoot apex and the leaves. So you have a delta of 2 hours in the wild type, and that delta is significantly reduced in the TCP19 mutant plants. It's not zero, so there might be other factors that also contribute to this, to basically create this time difference between the shoot apex and the leaf. And so the bottom line is, we go back to our model. Now we can kind of partially validate by this finding, this model, by the identification of TCP19 as the first organ-specific regulator of the clock function. Okay, so far, no other clock component has been shown to be, organ-specific. So what we're showing here is that the clock function, might have components that are really specific for one organ, and that are kind of fine-tuning the function of the circadian clock in that organ. There's still the big question. I'm sure I'm going to get that question of what this means, as I said before, and we have tried to come up with some hypotheses, and I'm going to show you one thing that we tested here. So one output of the circadian clock, as I mentioned before, is the regulation of flowering time. And flowering time requires for plants to properly measure day length, and plants measure day length by properly phasing the expression of certain genes to specific times of the day. Okay, and so we thought, well, maybe this phase change might have something to do with flowering. And so what we decided is to look at flowering time in the TCP19 mutant plants, and the flowering time experiments can be done in long days or short day conditions. Arabidopsis is a long day flowering plant, so the plants will tend to flower earlier in the long day condition. And in the TCP19 mutant, we didn't find any phenotype, okay? But what we reasoned is TCP19 is only regulating the expression of CCA1. But if we remember at the beginning of the lecture, or the seminar, what I mentioned to you is that CCA1 has a very close homologue LHY, which has very similar functions. And so what we decided was to see whether in an LHY mutant background, we could see an effect of TCP19 on the regulation of flowering time. And what we found is that at least in one of the conditions, the short day conditions, when we basically create a double mutant between TCP19 and LHY, which is this the last the picture of the last plant over here, you can see that there is a significant delay in the flowering compared to the two single mutants or to the wild type plants. Okay, so still we don't know the mechanism, but it seems like the one assumption or one hypothesis here is that this difference in phase or in peak between the shoot apex and the leaf is allowing the plants to properly measure and flower at the right time, at least in the short day condition. And so basically TCP19 influenced flowering time together with LHY. And so what I have shown you so far is that we started by basically not knowing very much about how the circadian clock might be, or the circadian system overall might be functioning in two different organs. And what we have found is that the circadian transcriptome and the associated biological processes to those genes that are rhythmic in one or the other organ are largely distinct and organ specific. We have also found that there is a common clock architecture. Okay, so clock components are conserved and expressed at similar levels in both organs that we studied. And we have found that there should be some amplitude modulation mechanism that we think it might be gene specific, that basically targets specific genes or specific sets of genes to make them arrhythmic in one or the other organ. And of course, finding those, I think, is going to be really challenging, because we're going to have to really target the right gene to be able to start dissecting how this mechanism might work. And finally, we found TCP19, as I mentioned, as the first organ-specific modulator of the clock function, which essentially, to some extent, at least partially validates this overall structure. And so, sorry, do we have 5 more minutes, or I can cut here? Yeah, what? Okay, sorry, guys. I'm going to take 5 more minutes to show you a little bit, a very small amount of data of another set of experiments that further validate this idea and this model. So one of the things, as I mentioned at the beginning, that or one of the aspects of circadian clock regulation that we're interested in the lab, is to try to understand how a local infection, affects the clock in distal tissues. And this comes from a previous paper from my lab that essentially showed that if you perform a local infection, what you can see is that in the infected leaf, there is a significant reduction of the rhythms overall, very, very rapid, okay? But what we were able to measure, and what interests us the most, is that if you look at the non-infected tissues, the tissues that were not in contact with the pathogen, you also see that the clock function is changing. And at that point, what we did was to look at all the uninfected organs, in other words, considering the shoot apex and the leaf together. And so the question here was, well, does this bacterial or single leaf bacterial infection can cause different changes in the distal tissues, maybe in the shoot apex, causing different changes as compared to the distal leaves? So we performed the same experiment, and we are just starting to analyze this, but I'm going to show you a little bit of the data. Well, we did the same transcriptomic analysis, but now what we did is to infect the plants before the collection of our time course, okay? So what we found very rapidly is that, again, the clock components don't seem to change. So in the infection context, the clock components seem to be expressed at similar levels on average, with similar amplitudes in both organs. Again, there are a few exceptions, but most of them, it's like the core clock seems to be unaltered, okay? Although we see that rhythms are compromised. Now, what we found is that when we look at the phase of the rhythmic genes, upon infection, we found that this is the same type of graph that I showed you before. The solid lines now are representing the data from the infected plants, and the dotted lines from the uninfected plants. And so the only thing that I want to point out here is that in the leaves, what we find is that the overall distribution of phases is very similar in the infected and the non-infected condition. But if you look at the shoot apex genes, there is a very significant shift or change in the timing of expression of these genes. Okay, this changes so much that now, instead of the genes peaking before in the apex and then in the leaf, this relationship is inverted. They tend to peak before in the leaves and then in the apex. Okay, and this is kind of represented here, where we see that there are major changes or more changes in the shoot apex, especially in the timing of gene expression, and not so much in the leaf. So to finalize, I would like to, basically show you this idea, which is, what can we predict from the model? What can be driving this phase difference between, one can be driving this very drastic change in the phases of gene expression in the apex and not the leaves. So if we follow the model, okay, and if we follow the model that indicates that phase or timing control is upstream of the clock, the idea is that what we would expect is that the entire clock should be shifted the other way around, okay, where clock components should be peaking before in the leaf and then in the apex. And that's exactly what we found. Okay, so this is the same type of graph that I showed you before, where before I showed you that most of the shifts were in red, showing that they were apex first. Now they're in blue, showing that they are leaf first. So to some extent, this is confirming, this overall, model where the timing of the clock is controlled up by mechanisms that are upstream of the clock. And then depending on what's the time of this overall clock structure, that's going to determine what's going to be the timing of expression for the genes that are downstream. Okay. And so just to finalize some very, broad idea here, what we are seeing is that the clock function seems to be A, highly conserved, and two, at least in the infection context, it also seems to be unperturbed, at least the core clock mechanism. And essentially, we find that there are two ways of tweaking or fine-tuning the clock function across different organs. One that controls the phase by mechanisms that are upstream of the clock, that controls the timing of the clock itself, and one that is downstream of the clock that ultimately we're predicting that controls the amplitude of rhythmic expression of the amplitude of the rhythmically expressed gene. And what we think is that this allows the plant to essentially respond to local environments. If you imagine, for example, a tree, that has, it's a very large plant, there might be parts of the plant that are being infected, parts of the plants that are under high light conditions or under different types of temperature and so on. And so what we think is that these different environmental signals might locally affect the clock function, okay, by tweaking, not by changing the clock itself, but by tweaking the mechanisms that are in the input or the output pathways in order to achieve or to allow that particular organ to respond properly. And so overall, what we think is that all these changes that are being fine-tuned are ultimately helping the plant to maintain, the overall organismal homeostasis. And we think that this essentially places the clock at the central, as a central component or hub of physiological control. Okay. So with this, I would like to finish just by some acknowledgements. I would like to thank the two students in my lab, one student that initiated the work in my lab, and the student that performed most of the work that I have shown you today, Nicholas Deason, and also Elsa Chen was part of this paper, and our collaborators at the Salk Institute and North Carolina State University, as well as our funding agencies. So thank you very much, and I'll be happy to take any questions.
Preguntas. Las personas en YouTube pueden escribir sus preguntas y aquí las leemos, por favor.
Thank you very much. Oh. Very nice talk.
Thank you.
And I have several questions, but well, first of all, it is very striking to think that in the what you call the shoot apex, there are expressed genes for flowering at that time, right? So you see that those ones are rhythmic, and they are rhythmic also in the shoot apex. That means that they
Yes.
are being expressed, right?
Correct.
So it's intriguing to me that flowering genes are highly expressed in the shoot apex. And my question is a specific type of flowering genes that are the ones that are expressed at that moment? Because, I mean, I would expect that flowering, once the shoot apex get into a flowering apex, you would start to expressing all those genes, but not before. And in this case, you are seeing it from the beginning.
Yeah, I would say that first, these plants are like 17 days old at the time, or 18 days old at the time of collection. You know, Arabidopsis will flower, in our hands at least, around 24 days.
Okay.
So the expectation is that the meristem will start changing way before the time you see the actual, you know, I mean, we count flowering as the inflorescence being 1 centimeter. So flowering happens probably at day 22 or even maybe earlier. Okay. So the first, I think, part of the answer will be, well, I think we're close to flowering. Okay. The second thing is that you need to keep in mind that these plants were moved to constant light conditions. And that's an inducer of flowering as well, right? Okay. So basically, by moving these plants, and these plants have been for 3 days under constant light conditions, so that can be a signal that is, you know, promoting, you know, the meristem to start changing to a flowering meristem. Okay. Now, in terms of the genes, we have looked at almost all the known genes that are participating in the pathway that controls flowering, and we have not seen any striking difference that might explain, you know, well, what the apex might be doing, or why, you know, it would be important for these genes to peak first in the apex and then in the leaf. Actually, because also, you know, flowering time, as you know, it starts by sensing day length in the leaves, right? And then FT is going to be translocating to the nucleus to basically induce flowering time. But we haven't found any striking. I know that we looked at them. We haven't found any difference to, you know, that's something that would call our attention, right? You know, in terms of being different than what we would expect.
And my second question is, TCP transcription factors are a family of transcription factors.
Yes.
And you find the 19.
Yes.
And my question is, are there other TCPs involved in this, or there are TCPs now in the leaf that might take function different to 19? Do you know anything about that? Yes.
Okay. So it's like I told you to ask me this question. Sorry. So CHE is a TCP transcription factor. Okay. So the first part of the answer would be, well, CHE is TCP21. I call it CHE for other reasons, but it's TCP21, and CHE is expressed everywhere. So that's I didn't want to emphasize too much that publication because it was kind of a long time ago, but CHE seems to be a transcription factor that regulates through the same site, right? The expression of CC1 in both the shoot apex and the leaf. I mean, now what's the relationship between CHE and TCP19? We still don't know. We're investigating that to see if there is some sort of partnership or heterodimerization of the two transcription factors. The next thing that you mentioned is, you know, could they be, or is it possible that there are TCP transcription factors that are unique to the leaf? Okay. So we didn't see them in the enriched genes. Okay, so at the levels that, I mean, we consider a gene to be enriched if the gene had more than 4 times, okay, expression in one organ compared to the other. Now, if it was 3.9, well, we didn't look at it, okay, in this analysis. So I think it's perfectly possible, and I think it's a very attractive hypothesis, right, that you're going to have some the same family of transcription factors, maybe through the same site, you know, maybe one activating, one repressing, something like that, and that basically contributes to also create the shift. Because, you know, we're talking about what I talked about today is that the shift happens because we're shifting the apex, but yes, you can also, you know, shift the leaf, so, you know, you create that difference, right? And so I think it is possible. What we're doing in the lab is now, you know, by using CRISPR to delete the TCP binding site, to start looking at, you know, what's the function of the site in controlling the clone function in the two organs, right? And that also, I think, is going to give us some indication on whether in the leaf there could be some TCP transcription factor that whatever provides different function. But even if it is Che, it could be that TCP19 and Che do something together in the apex, and then only in the leaves you have Che, where Che is going to do something slightly different. So even with the two that we know, we could start, building up some hypotheses, right? There are other TCP transcription factors, TCP22 and TCP20, I think it is, not from my lab, that also have been shown to control CCA1. So there is precedent for TCPs to control the clock function and CCA1 in particular.
And my last question,
Sure.
how do you think about modification, post-transcriptional modifications of the, some of these transcription factors, CCA1, or is it?
Oh, you mean
that it is
post-translation modifications of
right,
them, of the
right,
proteins?
Right.
Yes, I think it's perfectly possible. I mean, what I'm showing you here is, the clock, if we look at the mechanisms that control circadian rhythms, we're going to find that, transcription is one of the ways, we can control rhythmicity, but, protein stability, RNA stability, epigenetic changes, most of the most common ways of controlling ultimately protein expression or activity have been shown in one or the other organism to be associated to the clock function. So yeah, I think it's perfectly possible. And I think we are showing that at least at the transcriptional level, we can start explaining, how the clock works, but I wouldn't be surprised if, mechanisms like the one that you're suggesting also are having a role, right?
I'm going to ask you in Spanish.
Yeah,
right?
of course, yeah,
Okay.
sorry. Puedo tratar de responder en español también, sí.
No, sí, va, fuerza. Tengo la curiosidad de si se afectan funciones tan esenciales como la fotosíntesis en las hojas, que en parte pues son la identidad del órgano, ¿no? Y sobre todo en la etapa en la que usted lo está estudiando. ¿Qué pasa con el reloj en esas condiciones? Por ejemplo, una planta que, pues no necesariamente que sea albina, porque a lo mejor no llega a esa época, a esa etapa, pero que tenga afectada la función fotosintética.
O sea, en realidad lo que sucede acá es que la función fotosintética lo que cambia es el timing de cuando los genes que participan en fotosíntesis están en el pico, ¿no es cierto?
Pero no cambia el reloj.
No,
o sea, no cambian
perdón,
los
por ahí
genes.
no estoy entendiendo la pregunta, perdón.
Sí, no, a lo que voy es si la señal que viene arriba del reloj, que está definiendo la amplitud y el timing, puede estar dada por elementos que controlen a su vez funciones esenciales, como es la fotosíntesis, en el caso de
Seguro que sí, sí. El reloj, digamos, la forma en la que funciona el reloj, yo mostré este pathway lineal de input, clock, output.
Exacto.
En realidad es input clock, clock input, clock output, output clock. O sea, es un pathway bidireccional. Exacto. Donde los outputs que están siendo regulados por el reloj pueden feedback y cambiar la función del reloj, y lo mismo sucede con los inputs. ¿Okay? De hecho, la forma en la que el reloj controla, por ejemplo, sensibilidad a la luz a ciertas horas del día o defensa a patógenos, etcétera, esos son inputs que en realidad están siendo regulados por el reloj. Entonces la planta es más sensible, tiene un ritmo en la percepción de ese input, que está dirigido por el reloj. O sea que sí, yo creo que sí, que es muy posible que exista ese tipo de feedback loop, sin duda. O sea, lo traté de simplificar acá para tratar de seguir el modelo, pero sin duda es que la estructura del sistema circadiano es un reloj central, o de la forma en que yo lo veo como cronobiólogo, un reloj central que tiene una conexión bidireccional con múltiples pathways que regulan aspectos críticos de la biología y el comportamiento de la planta.
Entonces, en función de eso, la siguiente pregunta sería si no sería interesante buscar interactores de TOC o de TCP, en el sentido de que esa interacción que se debe dar en algún momento, ¿no? Pudiera definir entonces los genes que estarían cambiando su amplitud como usted dice.
Es exactamente un proyecto que tenemos en el laboratorio, está enfocado específicamente en eso y tenemos, no puedo decir los resultados todavía, pero sí tenemos resultados que indican exactamente lo que usted está proponiendo.
Una segunda pregunta es,
Sí.
en el caso de los últimos set de datos que nos enseñó,
Ujú.
la señal que se genera por el patógeno
Sí.
Es sistémica, ¿no? O sea, porque
Sí.
está afectando indirectamente o
Sí.
distancia la expresión en
Ujú.
el meristemo, en el
Ujú.
lazo. ¿Eso es específico de una infección por patógenos donde sabemos que hay una respuesta sistémica a los patógenos? ¿O podría ocurrir también en otras condiciones de estrés, ¿no?
Sí. Muy buena pregunta. Lo que publicamos en nuestro trabajo previo es que la señal es muy posible que sea reactive oxygen, no sé cómo se dice en español,
sí.
perdón.
Español.
Y de hecho, digamos, probamos con mutantes en una de las enzimas que produce peróxido de hidrógeno y vemos que está mediando esa respuesta, o partes de esa respuesta sistémica por lo menos. Y peróxido de hidrógeno, digamos, es común a múltiples estreses.
Claro.
Dice que si bien nosotros no hemos probado otros estreses, yo pienso que es exactamente, digamos, que lo que estamos investigando, que tiene que ver, yo creo que tiene que ver con peróxido de hidrógeno, porque además la capacidad o la fotosíntesis per se también produce peróxido, así que me parece muy atractiva la idea de que la planta está sensando niveles de peróxido que vienen del metabolismo normal de la planta o del funcionamiento normal y de todos los estreses a los que la planta puede estar expuesta, ¿no es cierto?
Exacto. Sí.
Entonces sí, no lo hemos hecho, pero si tengo que hipotetizar, yo diría que lo que estamos viendo acá seguramente se va a repetir si a esa hoja le aplicamos algún otro estrés.
Claro, sería muy interesante.
Lo que pasa es que he pensado por ahí, uno de los estreses que se puede aplicar, que puede llegar a ser fácil, es luz de alta intensidad. Temperatura no, porque es muy difícil en una planta de rebiosis solamente calentar una hoja.
Pero peróxido.
Pero peróxido sí hemos aplicado, peróxido aplicado a nivel, digamos, a nivel de toda la planta también, sí tiene un efecto. Tiene algunos efectos son los que vemos cuando aplicamos el patógeno. También lo hemos hecho con ácido salicílico, que también, digamos, es una hormona que está inducida por la presencia del patógeno y que también contribuye a la producción de peróxido de hidrógeno.
Exacto. Muy bien, muchas gracias.
Y perdón, para cerrar, los TCPs, también en otro trabajo hemos probado que los TCPs regulan la producción de la enzima, que se llama ICS1, que básicamente cataliza un paso crítico en la producción de salicílico.
Exacto, sí, muy bien.
Muy bien.
Gracias.
Bueno, tengo unas preguntas. Está muy interesante este trabajo que están desarrollando. Y mi pregunta está en relación de entender cambio de fases entre sample y hojas. Para entender esto, ¿cómo se hacen estos experimentos? ¿Toman todas las hojas o toman primer par de hojas, segundo par de hojas?
Esta es toda la hoja, pero hojas verdaderas,
sí.
Pero toda la hoja.
Sí, es que justo podría ser explicación de tipo developmental. Y entonces sabemos que el tiempo en desarrollo afecta muchísimo estos procesos. Y puede ser que primeras hojas pueden comportar en una manera, pero el segundo par de hojas puede comportar en otra manera. Y entonces quizás este developmental time es un aspecto importante. No trabajaron sobre esto.
No hemos trabajado, me parece una pregunta interesante, muy interesante. Son complicados estos experimentos para hacer la disección, etcétera, pero sí. Creo que puede ser totalmente. De hecho, hay algunos trabajos previos que indican que incluso los ritmos dentro de la hoja también tienen su variación. Pero bueno, decidimos empezar, digamos, por el órgano general y para después empezar por ahí a ir un poco más quizás más al detalle, más en profundidad. Pero bueno, antes de este trabajo, hasta donde yo sé, nadie había hecho una comparación en la misma planta tratando de buscar qué genes son rítmicos en una parte y en la otra. Sí, ahora tenemos como en cada time point tenemos las mismas plantas, las mismas cantidades de plantas, y lo que vemos, digamos, por primera vez, creo yo, es esta diferencia tan drástica en los grupos de genes que oscilan o que son rítmicos en un lugar y en el otro.
Sí.
Pero puede ser que en la planta vengan de distintas partes o que correspondan a diferentes estadios, sin duda. Sí.
Okay.
Sí.
Y la otra pregunta en relación de resettings. Entonces,
¿De qué, perdón?
de reset. ¿Cómo hacer reestablecimiento de tiempo?
Sí.
Por ejemplo, todo este trabajo está alrededor del ritmo circadiano. Y si hacemos un día de 12 horas nada más, con 6 horas de luz y 6 horas de oscuridad. Entonces, ¿cómo va a estar este resetting toda esta maquinaria transcriptómica que está actuando en un periodo y en otro y así? Entonces, mi pregunta es en realidad, ¿cómo es tan factible hacer este resettings? Y esta misma pregunta está relacionada con otra pregunta, que el ritmo circadiano depende de luz y oscuridad, pero también sabemos que campos magnéticos de nuestro planeta afectan estos ritmos también y no sabemos mucho cómo. Entonces, primero también en este contexto de resetting, si es posible establecer diferencias entre ritmos circadianos específicamente y campos magnéticos.
Ok, déjeme ver si puedes resumir.
Cada pregunta, pero es todo con...
No, perfecto. En cuanto al resetting, me parece que puede ser muy interesante ver si por ejemplo, el resetting es de última lo que ajusta el reloj a la nueva condición, ¿no es cierto? En seres humanos, para los que no estudian plantas, cuando tenemos jet lag, lo que sucede es que nuestro reloj interno está en un tiempo que es distinto al ambiente, ¿no es cierto? Y lo que sucede es que hay un resetting del reloj para irlo poniendo a la misma hora a la cual estamos viviendo, digamos, en ese momento. Entonces me parece súper interesante estudiar, por ejemplo, si el resetting funciona igual en los dos órganos, porque eso también nos indicaría un poco cómo puede ser el mecanismo, ¿no es cierto?
Si los mismos genes.
Exacto. Lo que digamos, nosotros no hemos hecho ese experimento, para mí se puede hacer usando estas plantas que son cis-1 luciferasa, por ejemplo, y uno puede hacer experimentos donde mira la luciferasa, que es lo que hemos hecho ya, en el apex o en la hoja o incluso en otras partes de la planta, puede ser el hipocotilo, la raíz, etcétera, y empezar a ver cómo la planta puede resetear su función. Ahora, no lo hemos hecho a eso, lo que yo no lo hice, pero un colega hizo, fue tratar de entrenar las plantas en ciclos de 4-4, 5-5, 6-6, etcétera. Y lo que sucede es que mientras la planta está en el ciclo de luz-oscuridad, trata de tener un ritmo que lleva el ritmo que tiene el ambiente, sea 4-4, etc. En general la amplitud tiende a ser más baja, porque es como que la expresión no llega a subir hasta el pico, porque enseguida viene la noche de nuevo. Pero lo que es interesante es que si cualquier planta que son crecidas en cualquiera de estas condiciones, ahora usted las pone en luz constante, todas vuelven a tener un periodo de 24 horas. En un par de días, al principio están como desacomodadas, pero en un par de días todas vuelven a oscilar con un ritmo de 24 horas. Con lo que quiero decir con esto es que mi interpretación de ese resultado es que muy probablemente la network y el reloj en sí ha evolucionado para que toda la cinética de todas esas mecanismos y reacciones que de última dan el ritmo, funcionan o han evolucionado para tener esa cinética de 24 horas. Entonces, cuando no dejan de tener el input del ambiente, vuelven a oscilar con ese ritmo de 24 horas.
Sí, pero justo puede ser diferencia en campos magnéticos.
Totalmente, sí. Y lo de los campos magnéticos me parece súper interesante. No conozco a nadie que lo esté haciendo en plantas. Hace rato que lo vengo pensando, como le dije a Gustavo y a los chicos ayer, a veces uno tiene muchas ideas, pero bueno, después de las ideas, lo que uno puede realmente hacer en el laboratorio, hay un camino. Y lo otro es temperatura, ¿no es cierto? Nosotros hacemos temperatura para las plantas, es un input realmente importante, y digamos, nosotros todavía no lo hemos considerado en nuestros experimentos, donde se puede hacer entrenamiento con temperatura o entrenamiento con luz y temperatura, y después mirar el reloj. Sí, hay muchísimas preguntas, sí, muy interesantes para responder. Gracias.
Muchas gracias.
Alguien de YouTube que se identifica como Desarrollo Sostenible MX hace dos comentarios. Uno, que los ciclos circadianos han explotado exitosamente en agricultura en plantas como crisantemos y marigolds, que no sé qué sean. Y luego la otra, que es muy interesante encontrar a nivel molecular cómo se regulan estos mecanismos. Y luego la pregunta que tiene es que si los factores de transcripción que contienen elementos de respuesta a la luz en sus promotores interactúan directamente con CCA1 y ajá, o si son regulados por otros motivos y otros mecanismos.
Ok. Los factores de transcripción, los que digamos, hay factores de transcripción que son regulados por la luz, tanto a nivel de la expresión del factor de transcripción, a nivel de RNA o de proteína. Hay factores de transcripción que interaccionan con el promotor de CCA1, como hay un grupo de VHLHs, que son los Phytochrome Interacting Factors, por ejemplo, que tienen sitios de unión en el promotor, en los promotores de CC1 y LHY. Ahora, a nivel de proteína, la verdad es que no me puedo acordar, no me sorprendería. Creo que hay algunos de estos PIFs que pueden interaccionar con CC1 y los criptocromos también se ha visto que pueden interaccionar, que son los PIFs están relacionados con luz roja, criptocromos con luz azul. En ambos casos hay relaciones directas entre los receptores o los factores de transcripción que son activados por los receptores de luz y los componentes del reloj, ya sea a nivel de proteína o a nivel de la interacción con los promotores de los genes del reloj.
I have a very fast question.
Sure.
I was thinking that the plant or the organism has a clock that is for upstream regulation, right? Like, okay, I wrote it because
That's fine.
Okay. If we have a general clock upstream, like likely controlled by TCP19, shifting likely the organ-specific timing or shift phase of the cycle. If we knock out the TCP19 in the apex, for example, would we see that it reshift to the core or like leaf-like cycle? I don't know.
So that's what is shown here, right? So sorry about that, but so you cannot, I mean, we have not mutated the TCP19 only the shoot apex. Okay, so we just mutated TCP19 in the genome, so it's everywhere, the mutation. So it's not a knockdown that is specific for the organ, right? But if you mutate it, it happens exactly what you're predicting.
Yeah.
Okay.
My question was if it is one-time shift, or, for example, if we knock out it, this shift is maintained, or
it is maintained here. So this is I think that this is what you're asking. Okay. So but
yeah,
correct me if I'm wrong. So basically, what this is showing is over 5 days, right? You have 5 cycles over here. What you see is the shift represented by where the arrows are pointing at, right? And now, if you mutate TCP19, at least for the course of these, time course, the shift seems to be maintained. So you see that the 2 arrows are together here, 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 and so on. Okay. So at least for the time that we did the experiment for, right, for these 5 days, the shift is maintained. So it's not that you have a shift on one day and then it goes back or something like that. I don't know if I'm answering the question. So
yeah,
kind of, no.
kind
We
of.
can talk about it later if you want.
Yeah.
Okay, that'd be great. Okay, cool. Well, yeah, the thing is that, for an organ specific, because the gene is only expressed in the shoot apex, the idea, and I think that these data is showing that, right? So you mutate in the shoot, you mutate it everywhere, but this gene is normally only expressed in the shoot apex. So it doesn't matter if you mutate it in the leaves, right? Because it's not going to be expressed anyway. So basically, by mutating the gene everywhere, okay, ultimately what you're achieving is a knockdown in the shoot apex, because that's the only place, as far as we can tell, right? There might be other exceptions, but as far as we can tell, the only place where this gene is expressed. Okay, so in a sense, you are creating a mutant that cannot produce TCP19 only in the shoot apex. If TCP19 was expressed everywhere, right? Then I think that yes, you might want to do that type of experiment where you mutate only in one organ and not the other one, right? We could maybe mutate CCA1 in one organ, not the other, and see, how the clock in each organ responds, because CCA1 is everywhere, and that's something we're attempting to do.
Yeah. So it will be to do that kind of experiment. We need to identify some other TFs for CCA1 in each organ to find out how that those ones shift the cycle in each organ.
Yeah, you can do that. You can use that as a readout of the clock.
Yeah. Thank you very much.
Muchas gracias a todos. No sé cómo se llama.
Después a ver si te escribo, porque quiero preguntarte cómo hacen, porque nosotros tenemos construcciones de los órganos, toda la vida, porque entonces pero vamos a ponerlo más
esa tiene la posibilidad es muy grande, entonces a veces afecta la función de la proteína. Entonces nosotros no hacemos función de proteína. No, es que ese es el motivo en general que cambia la función de la proteína. Hay un paper que muestra PTRR7, que es un reloj, Eva Farré, y ella hizo eso para ver cómo la proteína cambia. Pero creo que, yo no lo he probado, pero creo que está hablando del Steven, probó de hacer funciones tipo 1 o otras proteínas. Lo que empiezas a ver es que
Recording stopped.
la proteína